Google it, pangita sa tanan sa balita. And kadtong mga credible sites, basaha. Nya ka may timbang-timbang.
The body's immune system is altered during spaceflight. This is due to many factors. Various immune cells travel through the bloodstream, searching for invaders.
Due to delays in the delivery of reagents. Genome sequencing is a system that determines the presence of a new variant.
In space, aspects of the immune system appear weakened, which could lead to infections. Other aspects appear overactive, which can cause allergy or hypersensitivity. Certain viruses, such as the virus that causes chickenpox, have proven to reactivate in microgravity. This doesn't mean the astronauts are sick, but is evidence of immune changes in astronauts that may precede disease. Research on ISS is continuing to characterize exactly which parts of the immune system are altered during spaceflight.
As I said, that depends. Makasurvive ba iniktakoy nimo? Mo na magsige na sila
There are two main things. First of all, we're interested in understanding how spaceflight alters the infectious disease risk because we want to keep the crew, the astronauts, safe and mitigate their risk for infection when they're flying. But likewise, it turns out through the studies we've done in my laboratory and with Dr. Ott, we have been able to show that there are aspects of that microgravity environment that cells encounter when they're cultured in spaceflight that are directly relevant to what those same microbial pathogens encounter when they're in your body during an infection. Well, it all got started several years ago when Mark Ott at the NASA Johnson Space Center called me up on the phone and said that the astronauts were immunocompromised during spaceflight. Now that means their immune system doesn't function quite as well as it does when they're down here on Earth on a daily basis. And this intrigued me because my background and career training was in microbial pathogenesis. In other words, I study how microbes cause disease, the cellular and molecular mechanisms that they use to cause disease in your body. By studying those mechanisms, we can then apply that knowledge to develop new vaccines and therapeutics and treatments to prevent you from getting sick from infection. So I realized we knew half of the equation. We knew that spaceflight altered the immune system, but we didn't know the other half of the equation. And the other half of the equation is also very important to determine whether or not you're going to get sick after you get infected. And that has to do with the virulence of the pathogen. Now, virulence means disease-causing potential. But nobody had looked at the effect of spaceflight on the disease-causing potential. So I immediately asked Dr. Ott what I thought at the time might be a very silly question, and this should always teach you, always ask questions. No question is ever silly, no question is ever dumb. So I asked him, I said, well, this is amazing. I said, we have to look at the effect of spaceflight on the infectious disease potential of a pathogen. Has anybody ever done that? And his response was, no, they haven't. So I immediately followed that question was, well, we have to do this. Can we fly an experiment now? That was my naivete because I didn't realize you had to go through a very long series of ground-based studies to validate that you really needed to use the spaceflight experiment to address your potential question. I said, okay, until we can fly, is there some way that we can mimic, some system that we can use to mimic, to culture these bacterial cells here in the laboratory on Earth under conditions which mimic spaceflight? So this is that special bioreactor that NASA designed that allows scientists like me to culture our cells in the laboratory in the conditions which both simulate aspects of the microgravity environment and simulate aspects of inside our human bodies. So we have been able to use this bioreactor to grow pathogens in the conditions and ways that they actually experience in their bodies, in addition to similar ways in spaceflight, and find new ways that they're causing disease in the body. For example, my laboratory, in collaboration with Dr. Ott's laboratory, has shown that when we culture pathogens in this bioreactor, especially salmonella, a major human foodborne pathogen, it causes disease not only differently, but it globally changes its gene expression profile. It becomes a better pathogen. It can cause disease more effectively. It's a more robust pathogen. And it can also be more resistant to being killed by stresses that your body normally challenges you with. So armed with this information, this new insight we've been able to provide about how salmonella can cause disease in the body, by culturing it under spaceflight-like conditions, we were awarded a grant from NASA, our first one, to fly a spaceflight experiment.
In our case,
So this is the experiment we were funded in a grant with NASA to fly. What is the effect of true spaceflight on salmonella gene expression and disease-causing potential or virulence? Now, one thing to keep in mind is that spaceflight, doing experiments in spaceflight is very different than doing them here on the bench on Earth. You have to think differently, and there's a whole lot of constraints that you're going to have to consider. One of them to think about is we wanted to study the effect of spaceflight on the infectious disease potential of a pathogen. Well, the problem is NASA at the time did not allow anything to be infected in flight. So you could only grow the bacteria up there and activate them and prepare them for the experiment, but the actual infections have to be done back down here. So it's something to keep in mind when you're designing an experiment. So astronaut time is incredibly valuable, and you don't get very much of it. So you either have to work with hardware that's fully automated, or you have to design your experiment so that the crew has minimal amount of time that they have to invest in doing your experiment. Although I will say, the crew is awesome. The astronauts always want to give extra time to do science. Your experiment has to be designed to minimize hardware volume, to minimize mass, and power needs. You need to really minimize the amount of volume that you're sending up in flight, and that can pose a challenge for your downstream analysis of your samples, because you have to have enough volume to get enough sample to be analyzed for both gene expression and virulence. You obviously have to minimize mass because size constraints matter. You want to fly as small a volume as possible. And power needs, because most of the power has to go to run the shuttle. So we were very fortunate that our experiments could fly just at ambient or room temperature, and the bacterial cells would be fine. And we actually had the astronauts performing manually our experiments for us, so we didn't need the hardware to be as much of a power drain as the shuttle. So what did we find from our spaceflight experiment with salmonella? Well, we found that spaceflight uniquely increased the virulence of this pathogen, and it also globally changed its gene expression profile. And the unique part of the change in gene expression profile is that the genes that were being turned on and off weren't being turned on and off in a manner that would be consistent with salmonella actually being more virulent, actually being able to cause disease better. So what this taught us is that salmonella is causing disease in a different manner than we can detect when we grow it conventionally on ground when we fly it at microgravity. So greatly removing the force of gravity unveiled new ways that salmonella is causing disease in our body.